aerobic and anaerobic respiration, which should be popular. So please do remember to subscribe. And if you found it useful, then please recommend this to your friends. Okay, so let's make a start. Any point throughout it, you can ask questions through the Q&A box that's at the top of your login screen. Hopefully you can all hear me loud and clearly, and we'll begin. Okay. So why do you need a heart? Well, the reason you need a heart is because of surface area to volume ratio. Now, as organisms get larger, they have comparatively less surface area in comparison to their volume. Remember, the volume is going to be consuming, for instance, the oxygen, which is going to be turned into water, and the volume is going to be producing carbon dioxide from the link reaction and the Krebs cycle, and that carbon dioxide has to be got rid of. Now, if you have a small organism, they have a comparatively larger surface area in comparison to their volume, and this is often shown by measuring the size of a cube. So if we have a cube of side length 1, then it's going to have a surface area of 1 times 1 times 6. Whereas if we have a volume, it's going to give you a volume of 1 times 1 times 1 cubed. Now that gives us a surface area to volume ratio of 6 to 1. However, if we repeat this with something of the side length 3, then we have 3 times 3 times 3, which gives us 27, and it gives us a surface area of 54. So this gives us a surface area to volume ratio of 2 to 1. So as we've increased the size of the organism, the size of the surface area that the organism has for exchange has decreased. Now, organisms which are very, very small, they don't need a separate transport system. So things like amoeba, very small, therefore diffusion will give you enough oxygen, which can be used for aerobic respiration and get rid of the carbon dioxide, because these amoeba are heterotrophs just like you. Now, as we get larger, we've got some other options. So, for instance, this is a flatworm, which by its name is flat. So it's got a large surface area, it's very thin, it's also ectotherm, it's got a low metabolic rate. So simple diffusion is quick enough, and no cell is too far from the surface to get enough oxygen. So that's a flatworm. Now, when we get to a larger multicellular organism, earthworms are still fairly inactive, and due to their shape, they've got a large surface area to volume ratio. However, the problem is that the cells in the middle of the earthworm are quite a long way from the surface, so simple diffusion is not enough. So the earthworm's got a respiratory pigment that collects oxygen. It's got a system of tubes to pump the blood around, and then it's got a thickened muscular tube, which creates differences in volume, therefore differences in pressure, therefore it pushes the blood around the earthworm. Now, this is your heart, rather attractive, not actually your heart, but it's a heart. And what we can see here is we've got tubes coming in, so a big tube coming in here. This is a chamber here, which is an atrium, and this is an atrium here. Large ventricle, and there's 2, remember, because we've got a double chambered heart here. And then these vessels on the outside are the coronary arteries, because your heart is a muscle like any other muscle. It needs to get oxygen, it needs to get glucose, it needs to get rid of carbon dioxide. So the coronary arteries are supplying that blood to the heart cardiac muscle, which is contracting and therefore changing the volume of the ventricles. So mammals have got a circulatory system that consists of blood and then tubes for the blood to flow through, then a heart, and then we need a respiratory pigment to increase the carrying capacity for oxygen. So ours is hemoglobin. We have a concept of open circulatory and closed circulatory systems. So insects have an open circulatory system, which is a bit like putting their organs in a bath of tissue fluid and then swooshing that fluid around it, that the fluid isn't retained within in vessels. The blood is pumped from the top, goes down to the bottom, and then circulates its way back up again. And that supplies, that moves the tissue fluid around. And remember, insects get their oxygen through tracheae and tracheoles, which are direct connections from the spiracles from the outside, so they get their oxygen by diffusion. So they don't have to connect a oxygen exchange surface, a gas exchange surface, to the inside of the insect by a circulatory system. If we then look at a worm has got a system with a pump. It's got a valve, another valve. Blood exits the pump through an artery, goes through a system of capillaries, which is where the exchange occurs, and then comes back in through a vein. Moving through this. So the advantage of a closed circulatory system gives you more rapid blood flow. You can direct the blood to where it's needed, where it needs to be used. And the way you direct the blood is by altering the diameter of the tubes which are supplying those muscles. So the way you achieve this is by constriction, which reduces the diameter of the lumen, or relaxation, which increases the diameter of the lumen of the smooth muscle in the arteriole. Now, it's a common misconception that amongst students that the muscle in arteries or arterioles in some way propels the blood, which it doesn't ever. What it does is it controls the volume of blood that can travel through it by altering the diameter of the lumen of the vessel. Okay, and it gives more efficient delivery of oxygen because you're pumping higher pressure blood to those areas, to specifically those areas. So here's some examples of some closed circulatory systems. So in fish, you've got a single circulatory system where blood comes into the atrium, then goes into the ventricle, then goes through the gill capillaries, and then comes out of the gill capillaries having been oxygenated. Gill capillaries, remember, large surface area because lots of gill lamellae, countercurrent flow if it's a bony fish, very efficient exchange surface. Then the blood flows round the body of the fish and supplies the oxygen, which diffuses out down its concentration gradient into the mitochondria, because that's where the oxygen is being consumed and turned into water. And then the blood flows back round again. So this is a single circulatory system because for every circuit that the blood does through the fish, it does one circuit through the heart. We compare that to a mammalian heart. We've got an atria, we've got a ventricle. Blood comes out, goes through the lung capillaries, which is known as the pulmonary circulatory system, gets oxygenated, but also loses pressure. When you force blood through the capillaries, you're forcing them through very, very small tubes, and those very small tubes have proportionately more edge than a big fat tube. So that reduces the pressure because it causes lots of resistance, and then the blood flows back into the heart, where we then pump it pretty hard around the body. So this is a double circulatory system. So single circulatory, blood goes only once through the heart and then to the gas exchange. Double circulatory system, it goes through the heart twice before returning to the same location of the body, because it has to go through the pulmonary and the systemic circulatory systems. So single circulatory, heart gills, body, heart. And a double circulatory system has got the pulmonary, which goes heart, lungs, heart, and a systemic, which goes heart, body, heart. Join the two together and we've got the double circulatory system. Now, one of the reasons why we need a double circulatory system is that we are endotherms. Therefore, we have a very high metabolic rate. We need a lot of energy because we're maintaining a higher body temperature than the surroundings. So our cells need a lot of glucose, need a lot of oxygen, and produce a lot of carbon dioxide. Not much point going through that, it's fairly obvious. Right, so the advantage of the double circulatory system is that the blood flows more quickly to the body tissues because it's been repressurized as it has gone back into the heart. So the oxygenated blood is at much higher pressure than it would be had it just, for instance, come out of the gills. So it's more efficient delivery of oxygen and nutrients, glucose. You supply more oxygen to the heart muscles because the heart muscle can work harder because it's getting more oxygen. because it's part of the systemic circulatory system. And then the systemic circulatory system can carry blood at a much higher pressure than the pulmonary circulatory system because of problems with squeezing plasma out into the alveoli. So with a single circulatory system, the blood pressure is reduced as the blood passes through the capillaries of the gills. Therefore, you've got oxygenated blood, but the blood flow, the velocity of the blood is not very quick. Therefore, you're limited in your supply of oxygen and glucose to the respiring muscles of the fish. So this is why the single circulatory system isn't as good as a double circulatory system that we have. Just a point of an odd one, some amphibia. So some amphibia have got a single ventricle and 2 atria. Now, how this works is you have deoxygenated blood coming in from the systemic circulatory system, and you've got oxygenated blood coming in from the pulmonary. Now, those two both mix together in the ventricle, and then when you pump the blood out, there is some oxygenated blood and some deoxygenated blood which will go through the lungs. And then there is some oxygenated and some deoxygenated blood which will go around the systemic capillaries. So amphibia have got a mixture between the two, whereas birds, mammals and most reptiles have got a double circulatory system. Okay, different parts of the heart. So now if I just bring up a whiteboard, I've got a couple of points I'd like to make. So when you've got a heart, it's a thickened, some muscle around a vessel. It's very important when understanding the heart to understand the principles of what's going on, is that when the muscle contracts, the volume of the chamber decreases. Now, when the volume decreases, the pressure increases. And when the pressure increases, blood will move to where there is a lower pressure. Now, we can increase the efficiency of this system by putting in 2 completely passive flaps of skin. And one flap of skin is here and the other one is here. And these passive flaps of skin we call valves. Big misconception with students is that valves are permissive, that they open and allow the flow of blood through. They are pushed open by the flow of blood and pushed closed by the flow of blood. They don't open and allow the blood through. It's a subtle point, but very, very important, particularly when you come to exam questions. So now when the heart contracts, then blood will be pushed towards this totally passive flap of skin, which will be pushed closed. So blood can only exit in one direction, which is this way, which would be towards the artery. When it does, the pressure will increase, so that will push the artery walls out. And then when the ventricle has stopped contracting, it will start to relax. At that point, the pressure will be higher where the blood has been forced into. So the walls of the artery will contract back because the walls are elastic. So they're going to push back onto the blood. So it's going to push the blood back towards the chamber, so back towards the ventricle. Now, as it does that, we can increase the efficiency again by putting in another set of passive flaps of skin. So now, when the pressure is higher in the atrium, where I put the the artery, where I put the crosses, the blood will be pushed back, and that will push the flaps of skin closed. So now blood can only carry on going this way around your body. So again, to repeat the principles, when the volume decreases, the pressure increases. Blood will move to where there is a lower pressure. As it moves, it will push valves open, and then the valves will be pushed closed by the blood flowing back to where it was before. Okay, going back. So some of the chambers for you, you've got the vena cava, which comes in, and this is coming into the right atrium. You've got the pulmonary vein, which is coming into the left atrium. you've got the atrioventricular. I'd like the term atrioventricular because it tells you where it is. It's between the atrium and the left ventricle. You've got the atrioventricular valve. You've got a left ventricle which gets the blood from the pulmonary circuit and then pumps it out around the systemic circuit. You've got an aortic valve between the aorta, which is the blood vessel leaving the left ventricle, and then you've got the aorta, which is taking the blood around the systemic circulatory system. The septum is this bit down the middle, which divides the two parts of the heart. And then you've got a pulmonary artery, which is supplying deoxygenated blood to the lungs. You've got a pulmonary valve, and then you've got the right ventricle and the tricuspid valve, which is the valve between the right atrium and the right ventricle. Again, I like the term atrioventricular valve because it tells you where it is. A point of these things here, these are the chordae tendineae, or tendinous cords. And the reason why you have these is because the pressure difference between the ventricle and the atrium is considerable. And if they weren't there, then the valve flaps could be pushed back into the atrium. But by having these chordae tendineae, it restrains the valve flaps. It means they can only move a certain distance. Now, you don't have chordae tendineae on the aortic or the pulmonary valves because the difference in pressure between the pulmonary artery and the aorta and the left ventricle and the right ventricle is not as great as between, say, the left ventricle and the left atrium. Okay, I hope you're still with me. So we got the mammalian heart's a pump for both sides. The heart muscle is myogenic. That means the impulse to contract comes from the heart muscle itself, although it is coordinated by one part of the atrium, which we're going to look at later. Atria's got a thin muscular wall, which exerts a small pressure to push the blood through to the ventricles, and it pushes through the atrioventricular valve, which is already open when it contracts, and we'll look at that in a moment. Right ventricle has to push the blood around the lungs, so it's thicker than the atria. Left ventricle has got to achieve the highest pressure, so it means that because it's got to pump the blood from the left ventricle into the aorta, and the pressure in the aorta is much higher than in the pulmonary artery, so that the thickness of the muscle needs to be up to 3 times thicker than the right hand side, because it's got to achieve much higher pressures to pump the blood out into the aorta, because the systemic circulatory system is at a much higher pressure than the pulmonary circulatory system. Okay, the reason for the septum is it stops the mixing of the deoxygenated and the oxygenated blood, and it allows different pressures to be maintained in each circuit. Okay, let's talk through some of this. So this might work. No, it's not going to work. Right. It's unfortunate, but there we go. Let's keep going. Okay, so blood comes into the atria. The atria contract, blood is pushed down. The ventricles contract, and blood is pushed back towards the valve and pushes this valve closed between the atria and the ventricle, and then pushes blood out into the arteries. Now, point here is that when the atria contract, their volume decreases, their pressure increases, and blood goes from a higher pressure in the atria to a lower pressure in the, hang on, just get me, there we go. So it goes from a higher pressure in the atria into a lower pressure in the ventricle. Bloods come into the two ventricles, then the ventricles contract, the pressure increases, and when the pressure is higher in the ventricle than it is in the aorta and the pulmonary artery, then blood gets pushed into the aorta and the pulmonary artery. So remember the principles here. The volume decreases, therefore the pressure increases, therefore the blood flows to where there is a lower pressure. Okay, so how valves work? Valves are pushed open when the pressure on one side of the valve is higher than the pressure below it. The atrioventricular valve will open when the pressure in the atrium is greater than the pressure in the ventricle. The semilunar valve, or aortic or pulmonary valve, will open when the pressure in the ventricle is greater than the pressure in the artery on the other side. They're pushed closed by the backflow of blood when the pressure beneath the valve is higher, and so on. Blood flows back towards the atria or the ventricles and then force the valve shut. Okay, the sound you hear when you listen to somebody's heartbeat, the lub-dub sound, is the closing of firstly the atrioventricular valve, and then the dub sound is the closing of the semilunar or aortic or pulmonary valve. Let's skip this exam question. Right. Now this graph is something that causes a lot of consternation and stress among students, and I can understand why. So let's just do it on a whiteboard and we'll try and build up the concepts of what's going on. So that's a graph of the pressure changes that occur in the heart. So, and then along the bottom we have time. So we put time here and pressure in kilopascals here. Now when you get this graph and you've got to talk through it, first thing to remember is to draw yourself a little heart on the side. So you draw an atrium, a valve, a ventricle, and a valve, and then an aorta. Now, having drawn that on the side, that gives you a few things to think about and to help you structure your questions, structure your answers, sorry. So if we start with the atria empty and we do the atria in a blue line, the atria are going to fill up with blood because blood is being returned back into the atria. And if we're talking about the left-hand side, it's going to be returned back in through the pulmonary vein. So as the blood's coming in, the pressure's rising and rising and rising in the atria until there is a higher pressure in the atria because of the build-up of blood that's returned from the pulmonary circuit. And that pressure is then high enough that it pushes open the atrioventricular valve, at which point the pressure will fall in the atrium because the blood will then go into the ventricle. So we then see a fall in pressure when the atrioventricular valve opens. If we then go back to 0 time, so this might be here, it might be 0.8 of a second. If we go back to 0 time, what happens next is the atria contract and the pressure goes up. So with the cycle with an atria, we've got increase in pressure here, 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 because the blood's coming back from the pulmonary vein. Then you've got a fall in pressure because the blood has gone from the atria into the ventricle. And then you get a rise in pressure because the atria contract and push the rest of the blood through the already open valve into the ventricle. Okay, now let's consider the pressure in the ventricle. So the pressure in the ventricle will rise because blood is being pushed in from the atria, and then the ventricle will contract. Now, when the ventricle contracts, the pressure goes up because the volume's decreasing, therefore the pressure is increasing. Now, the blood that's in here, as the ventricle contracts, will push the blood back towards the atrioventricular valve, which will push the valve closed at that point. So where the line crosses is where atria and ventricle, where the valves open or close. So at this point, the atrioventricular valve will close. Because the pressure goes up in the ventricle, becomes higher than in the atrium, the blood is pushed from the atrium, from the ventricle back into the atrium, which pushes the atrioventricular valve closed. Now, you've then got to consider the pressure in the aorta. Now, the pressure in the aorta is very high. So the pressure in the aorta will prevent the aortic valve from opening until the pressure in the ventricle becomes higher than the pressure in the aorta. So only at this point here will the aortic valve open. When the aortic valve is pushed open by the higher pressure in the ventricle, then blood will be pushed from the ventricle into the aorta, which will cause the pressure to go up. Now, as that happens, the pressure in the ventricle also goes up because it's still contracting. It's forcing blood out, and then the ventricle begins to relax because it can't contract forever because it's a chamber, it's contracted inwards. And as it relaxes, the pressure begins to fall. And as the pressure begins to fall in the ventricle, then the pressure also begins to fall in the atrium. Now remember, sorry, not in the atrium, in the aorta. Now remember, we've pushed the sides of the aorta out by the increase in pressure caused by forcing the blood out from the ventricle into the aorta, because the pressure is greater in the ventricle than it is in the aorta because the heart's contracted. Now, these walls are elasticated, so these will then push back onto the blood, which then forces the blood back towards the aortic valve. So the pressure is going to fall in the aorta until it becomes higher in the aorta than in the ventricle, because the pressure in the ventricle is falling quicker because the ventricle is a chamber that has contracted and is now relaxing. So then when you get to a point where the pressure in the aorta is greater than in the ventricle, then at that point, the aortic valve will be pushed closed by the flow of blood. So the aortic valve is only open from this part to this part. So that's when blood is being pushed out of the heart because the pressure in the ventricle is great enough to push it out into the systemic circulatory system. Now then we've got the pressure falling in the aorta and falling in the ventricle and falling in the ventricle and falling in the ventricle, falling in the ventricle because the ventricle's relaxing until the pressure becomes lower in the ventricle than in the atrium. So at that point, the atrioventricular valve will open. So remember the principles. When the blood moves from a higher to a lower pressure, the reason why the pressure in the atria goes up at this point is because blood is being returned from the lungs in the pulmonary vein. When it's higher than it is in the ventricle, the valve gets pushed open. The atrium then contracts, which pushes the rest of the blood through the already open valve. Then the ventricle contracts, which pushes the atrioventricular valve closed. The ventricle continues to contract and both sets of valves are closed, and it's squeezing the blood until the pressure is high enough to push the blood out through the aortic valve, goes into the aorta, causes the walls of the aorta to expand outwards. The ventricle stops contracting, begins to relax, and then the blood will start to flow back from the aorta towards the ventricle, which will push the valve closed. Then the ventricle will continue to relax until it's lower in pressure than in the atrium, and because the atrium's been filling up with blood. Now, contraction of the heart is called systole, and relaxation is called diastole. And I see I've got a question. Why is the atria contracting at the start and not after 0.8 of a second? So Mo, it is contracting after 0.8 of a second. This is one heartbeat. So this is atria contracting and then filling with blood and then contracting again. But the reason I start here is I think it makes more sense to explain why this is filling with, why the pressure is rising here until the pressure becomes high enough to push the atrioventricular valve open, because I think that point gets lost on some students. So that's something I wish to emphasize by approaching it that way. Okay, I hope that made some sense. clear this and go back to so here it is. This is your heartbeat. So, and the heartbeat going from 0 seconds to here, this would be 1 heartbeat going this length, and then it would be starting again here. Now, if we were to look at this as what would be happening on the right hand side, then the right hand side would be exactly the same, except it would be a considerably lower pressure in the pulmonary artery, because the right ventricle is not as thick as the left ventricle, so that the pressure would be much, much lower, and therefore the pressure would be lower in the pulmonary artery. So the whole system is exactly the same, just at a considerably lowered pressure. Okay, so if we add on to this some of the volume changes in the ventricle, then you can see that the volume decreases here as the pressure goes up. And then this is the electrical activity, which we're going to look at a bit later. So the P wave is atrial systole, so atrial systole occurring here. The QRS wave is the electrical activity associated with contraction of the ventricle, and then the T wave is ventricular recovery. Okay, now's your time to ask any questions, and I'll load up the next quiz. So the next quiz is running now. So hopefully you should be able to have a go at some of those quiz things. It's on in the Socrative room. So if you go to Socrative, put in A-level biology tutor as the room code. Got a chat point coming up. What's the best way to revise this topic? Keep in mind the principles. Keep in mind the principles of why the blood is flowing, which is the changes in pressure, why the pressure is changing, which is the changes in volume, and why the valves open or close, which is the relative pressures on either side. More questions. What's the difference in ventricle pressure on the graph when comparing the left and the right hand side? So I don't know the exact pressure of the right ventricle, but it's considerably lower because there's a big difference in the thickness of the left and the right hand sides. They never ask you to say what the pressure is, but they always ask you to annotate a graph with what the pressure would be, trying to make sure that you make the point that it would be exactly the same curve, just be lower because of the lack of thickness of the heart muscle of the left compared to the right compared to the left. Hope that answered you. Okay. Any more questions, please feel to ask them. Hopefully you should be doing the second Socrative quiz, which is at socrative.com. Emma, you've raised your hand. I'm not quite sure what that means, but if you'd like to ask a question, then you're very welcome to in the Q&A box at the top. One in the chat room. Sorry, that was an accident. Oh, well, Emma, no problems. So I've only got about half of you in the Socrative room. So it's, and one of you's done all the questions. So if you're unfamiliar with this, remember you go to socrative.com, go to students, put in A-level biology tutor in the room number, and then it takes you straight to the quiz. And there's 5 or 6 questions which you can do, multiple choice questions which are there to reinforce your understanding of the topic. So do go along and get involved in the quiz. A couple more of you in the quiz room. A few of you have finished, which is great. Well done. Got another question. How often does the surface area ratio tie into the heart and hemoglobin affinity? Not quite sure where you're coming from with that, Chloe. The surface area to volume ratio normally comes up with the need for a gas exchange system. So they would talk about that with why do you need gills, or why do you need some irregular shape, or why do you need to change the shape of the organism so it's flatter or longer or thinner in order to increase the surface area to volume ratio. The reason why I started with surface area to volume ratio is that it's one of the points that students tend to miss, is that it's as a result of the decrease in surface area is why you then need a transport system to connect the exchange surfaces, because transport systems are connecting an exchange surface in the lungs or the gills to an exchange surface which are the capillaries in your muscles, or the muscles of the fish, for instance. So the heart and the blood vessels connect those surfaces so that one surface can receive oxygen and one surface can get rid of the carbon dioxide and pick up the oxygen. So the reason I started with that was to try to make that point. I hope that answers your question. Any other questions, people? Please feel free to ask it in the Q&A box. Well done to everybody who's finished the quiz. What's the difference in the arteriole and the artery? Well, the difference is the thickness in the wall and the size of the lumen. An arteriole is a vessel that comes off the artery, a bit like an A road coming off a motorway, and it's a smaller lumen with a thinner wall because it's at a lower pressure. And the reason the pressure is lower is because there's more resistance, because there's more edge, but we're going to talk about that in about 2 minutes, 3 minutes time. Hope that answered your question. Any other questions, people? Okay, in that case, I will move on. So if we look at now the control of a heartbeat, and then we're going to look at arteries, veins, and capillaries. So we start with the heartbeat starts off in the sinoatrial node, and you get a wave of contraction that spreads over the atria. So remember, the atria are contracting and pushing the blood through the already open atrioventricular valve. Now, then you've got a layer of connective tissue which is between the atria and the ventricles. Now, this layer of tissue insulates the atria from the ventricles, which means that the electrical activity that's coming out from the sinoatrial node doesn't then spread down and cause the ventricles to contract at the same time, because there needs to be a pause for the blood to be pushed from the atria into the ventricle before the ventricles contract. Additionally, remember that the blood is doing a U-turn. So the blood that's moved in is going to exit through the aorta and the pulmonary artery, which are at the top of the heart, so the top of the ventricles. So the blood that's being pushed through from the atrium into the ventricles is then going to exit from the top, and that requires the ventricles to contract from the bottom up. So you've got to achieve two things here. Firstly, the sinoatrial node causes the atria to contract. Then it gets picked up by the atrioventricular node. Now, the electrical activity passes quite slowly through the AVN. It takes 1/10 of a second to pass through the atrioventricular node. And that's slowing it down, and then it travels down the bundles of His, and then that causes the ventricles to contract from the bottom up, therefore pushing blood out of the top. So heart muscle, remember, is myogenic, is that the impulse to contract comes from the inside the muscle itself. And it's, I think, due to the permeability of the muscle to potassium, so it's constantly causing action potentials. I cannot quite remember, but there we go. And then we've got sinoatrial node, which is the pacemaker, which is then coordinating that heartbeat. So again, going through, we've got signals coming across from the sinoatrial node, which is in the right atrium, causing the atria to contract. Now, you can detect this wave of electrical activity on an ECG, and this is called the P wave. And the P wave is the electrical activity spreading out from the sinoatrial node, causing the atria to contract. Now, this small pause here for 1/10 of a second is the pause at the AVN for 1/10 of a second. Now that enables blood to be pushed from the atria into the ventricle, and then from the AV node it travels down the bundles of His, and then the contraction of the heart starts from the apex. So then we see the QRS wave, and here QRS wave, which is the electrical activity caused by the contraction of the ventricles. And then we have the T wave, which is the bump afterwards, which is the recovery of the ventricles. So again, making those points, we've got a P wave, which is atrial systole caused from the sinoatrial node, QRS wave, which is the contraction of the ventricle, and the T wave, which is the recovery of the ventricle. Should we have a look at this or not? Maybe there's a good point to this. Yeah, perhaps there is. Right, so at what time do the semilunar valves close? Well, the semilunar valve closes. The semilunar valve is between the aorta and the ventricle, so the semilunar valve will close here. And then calculate the heart rate in beats per minute. What you need to do is to take this to this, so it takes 0.8 per second, so you divide 60 by 0.8 per second. Oops. So there you go. The semilunar valve closes at 0.4 of a second, so we can read it off here. And then to calculate a heart rate, you look at any 2 points where the same point exists. So you could go from here to here, or you could go from here to here, and then divide 60 by that time, which gives you the heart rate in beats per minute, which in this case is 75. Right, on to blood and blood vessels. So what's in blood? So I'm not going to do hemoglobin and dissociation curves, because that can be something for another lesson, because it's a full-on big topic when you consider it's got to do ball shift and fetal hemoglobin and dissociation curves. So I'll do that on another topic sometime in the future, if enough people come to any more of these revision sessions, which hopefully they will keep coming. Okay, so what's blood made out of? Well, we've got plasma, which is water and dissolved salts and the products of digestion. You've got white blood cells and platelets, which do the clotting response and the immune system. And then you've got erythrocytes, which are red blood cells. And in you, the number of erythrocytes you have, the hematocrit level is about 40 to 45%. If you're a Tour de France cyclist on enough EPO, then I think the highest they've ever recorded was about 62%, which is quite astonishing, and attributes to their ability to force what is virtually jam down their blood vessels. Okay, here are some erythrocytes. Remember, there's no nucleus, and whereas here and here, these are some white blood cells. Okay, so role of blood. It's a transport medium. It's going to distribute heat from organs which are net producers of heat, such as the liver. It's a buffer. It's going to buffer the pH because it accepts hydrogens to, when you've got transport of carbon dioxide. And that's in a future lesson. And it's got a defense system because of the clotting response and also the presence of the white blood cells. And it provides pressure for some organs to function, namely the kidney in particular, where it's got to generate a pressure to give you a glomerular filtration rate where you push the plasma from the glomerulus into the Bowman's capsule. Okay, arteries take blood away, veins return low pressure blood to the heart, and capillaries connect the arteries with the veins. Blood vessels, the arteries and the veins all have the same structure, as do the arterioles and the venules, except for the capillaries, which are only a single layer of cells. All of the others have 3 distinct layers of cells. Where they differ is in their relative thickness, depending on the function of the vessel. So around the outside, we've got tunica externa, then we've got tunica media, and then you've got tunica intima or tunica interna. And then this is the lumen through which the blood flows. So tunica externa, lots of collagen, which is a strong fibrous protein. Tunica media, we've got smooth muscle, which definitely does not propel the blood under any circumstances ever. The reason for the smooth muscle is that it can constrict and reduce the lumen. And when you can do that, you can then control where the blood is flowing to. So for instance, with vasoconstriction and vasodilation, which are responses by endotherms to changes in external temperature, then you can, if you're too hot, you can vasodilate the arterioles going to your skin, and therefore you lose more heat from your skin. And the reverse is true if you get too cold and you want to reduce the heat loss from your skin. You've got elastic tissues, which allows the artery to stretch, and as it recoils back, that's going to help maintain the pressure. And then we've got tunica intima, which is a single layer of endothelial cells, which are very smooth and very flat to allow the blood to flow with less pressure, with less friction. Okay, so here's the comparison between an artery and a vein. So the big difference is, hopefully you can see here, is the thickness of the tunica media. So you've got a big thick tunica media here in the artery and a much thinner tunica media in the vein. Now, additionally, there's a big difference in the size of the lumen, because remember, the pressure is extremely low in the vein. So the only way you achieve reasonable velocity in the vein is by having a very big lumen, because if you've got a very big lumen, you've got comparatively less edge to generate the friction. I think edge, I don't know whether edge is the best term, but it's the term I'm going to use. So with an artery, you've got high pressure blood flowing down a lumen, which is much narrower than the vein, but the vein has to have a much bigger lumen because it's at much lower pressure. Both of them have got a tunica externa, and the tunica media is the big difference between the two. And then the veins have valves and the arteries do not. Between the arteries and the veins, we have the capillaries. The capillaries are only one cell, one endothelial capillary and endothelial cell around the capillary, so that gives you a very short distance for diffusion. The capillaries are very narrow, which is going to cause a decrease in the pressure, because it's going to cause an increase in the resistance, because you're forcing blood through a very small tube, and there's lots of them. Okay, so here's some cross sections through some arteries and some veins from Roland the rat with the donor card. This is the vein, and here we've got a comparatively thin tunica media, and this is an artery, and we've got a much thicker tunica media. Another couple here. Here's another artery, much thicker here and here, and again the vein much thinner here and here. And this here is a capillary, and running through the capillary you can see the red blood cells, which are the darker blobs there. Just to give you an idea of a sense of scale of just how thin the capillaries are, they're about the diameter of a single red blood cell, which is pretty small. Again, vein, artery, difference in the tunica medias. Artery wall with tunica externa here, which is the collagen, tunica media, which has got the smooth muscle, the elastic fibers, and then tunica intima, giving you smooth flow. Okay, so the arteries take the high pressure blood away from the heart. You've got thick walls, you've got a narrow lumen to maintain the pressure, lots of elastic tissues to enable it to stretch and then recoil back on it. And you feel a pulse running through you, which is the recoil of the blood as it's being pushed through your system. And then the arteries can change their diameter by contracting the smooth muscle or relaxing the smooth muscle to supply more blood to certain areas. So if you go to the gym and you do lots of bicep curls, then your biceps will become filled with blood because your arterioles will relax going to your biceps, which will cause your biceps to have more blood going to it because they need more oxygen and to take away more carbon dioxide. So capillaries, I quite like this picture. I mean, these are red blood cells here stacked up inside a capillary. So you can see them running through here, which I think is quite nice and gives you an idea of the relative size of the capillary and the red blood cell. Now, point that students tend to miss is that because you've got a very narrow diameter of the lumen, it means that you have more wall, more edge in comparison to a big fat tube. Because of that, you've got more friction. Because of that, you slow down the flow of blood. Therefore, the pressure is lower. Now, as you've slowed down that blood, it means that you can then have more exchange of oxygen and carbon dioxide, because the carbon dioxide is going to be diffusing out of the mitochondria and into the red blood cells, where it's obviously going to be turned into hydrogen carbonate with carbonic anhydrase, and then into hydrogen ions with the whole transport of carbon dioxide thing. And then the oxygen is going to be diffusing off the hemoglobin down its concentration gradient into the mitochondria, because therefore there it's being used to make water in oxidative phosphorylation. So you got the diffusion of oxygen one way, carbon dioxide the other way, and obviously the diffusion of glucose as well, tissue fluid, which is something for another lesson. Okay, functions of the capillary. A single cell thick, which gives you a short distance for diffusion. Small diameter of the lumen gives you friction and slows the blood down. Slowing the blood down, which means you've got a better chance of exchange. And you've got a very large number, so this gives you a large surface area for the exchange of oxygen and carbon dioxide and blood plasma. Okay, on to veins and venules. So let's get my pointer thing. Okay, so we've got an artery here, we've got a vein here, and you can just see the size of the lumen in comparison to the thickness of the wall. So the purpose of the vein is it returns low pressure blood to the heart, much thinner wall, massive lumen, and the reason for that is that when you've got a big lumen, you've got less edge, so you've got less resistance to blood flow. Outer layer of collagen, little elastic tissue because it doesn't have to resist great big pulses in blood and maintain the pressure because there isn't any. And then it's got valves to prevent the blood flowing back to the heart. No, sorry. Yes, prevent the blood flowing back towards the capillary and then back to the heart because you want the blood to keep going forwards to the heart. Now, summarizing that again, little elastic tissue because it's at low pressure, large lumen returning the blood back to the heart, therefore less edge, and valves to prevent the backflow back towards the capillary because you want it to flow onwards to the heart. Okay, so how does the blood get back to the heart? So this is a source of some confusion with students, and it's worth making these points now. So from organs above the heart, it gets there by gravity. So it's going from anything above your heart down by gravity. From organs below the heart, and this is where it causes a bit of confusion, is the action of the skeletal muscle. Now, the skeletal muscle is not propelling the blood through the veins back to the heart. It is the skeletal muscle is the muscle that you use when you're walking around. Now, when you walk around, those muscles contract, and when they contract, they expand. And when they expand, they put pressure on the tubes that are running through them, which may be the veins. Now, when they put pressure on the veins, they're going to reduce the volume of the vein, so it's going to squeeze the blood, and that will push blood through valves, keeping it going towards the heart. So when you say the action of the skeletal muscle, it's the skeletal muscle that is moving you around, or moving your arms or moving your legs, and that is then squashing the veins that go through your run through those muscles, and then that's causing the blood to be pushed through the valves and around the rest of your body. Additionally, when you breathe in, you move your diaphragm down, which reduces the pressure in the thorax because you increase the volume of the thorax. When you increase the volume of the thorax, you reduce the pressure, and your heart is in that box that you are increasing the volume of, therefore reducing the pressure. So when you breathe in and move the diaphragm down, you're not only helping air move into your lungs because you're increasing the volume of the box that the lungs are in, but you're also increasing the volume of the box that the heart's in, therefore the blood, that helps with the return of the blood back to the heart. Okay, a little bit on valves. Blood goes through the valve, blood comes back and pushes the valve closed. That's it. Skeletal muscle, as you walk around, the muscle contracts. As the muscle contracts, it's going to squash the blood. As it squashes the blood, it's going to push that blood. It's going to squash the blood like this. The pressure is going to increase here. The blood's going to be pushed back towards the valve, and it's going to push the valve open to continue its journey back to the heart rather than towards the capillaries, which is the wrong way. Okay, now pressure changes in the systemic and circulatory system. So this is a graph which can cause a bit of angst amongst students. So we have these fluctuations here in the systemic circulatory, in the pressure in the systemic circulatory system, and this is due to the contraction of the heart. Now, if we think about it really hard, this is actually just an extension of the graph we looked at before. So as the pressure increases in the ventricle, when it gets high enough, it pushes blood out from the ventricle into the aorta, which causes this rise in pressure in the aorta. I'll just clear that off. Now, as you go along the aorta then splits into arteries and then into arterioles. And it's going into more and more tubes, which are narrower and narrower and narrower tubes. But there's more and more of them. So the total cross-sectional area of the tubes is increased, but the amount of edge which creates the friction is also increased. So this is what's starting to cause this fall in pressure. Then it goes into more, even smaller arterioles, which again means it experiences more friction, therefore, and a larger total cross-sectional area, therefore the pressure falls more. Then we've got the start of the capillary and the end of the capillary. Remember, the pressure is higher at the start because there's a higher hydrostatic pressure, and also then you then force plasma out, forming tissue fluid. As you go along the capillary, remember the capillary is a very narrow tube, so that's going to cause, again, a drop in pressure. And then the pressure is much lower in the venules and in the veins. And then it goes into the pulmonary circulatory system, which has got a much lower pressure because of the relative thickness of the right ventricular wall. Same pattern though. And then around the capillaries in the lungs, and then back through the pulmonary vein, back into the left-hand side, and then again the fluctuations in pressure from there. So the pressure falls due to the resistance and due to the friction with the vessel, walls of the vessels, and plus there's a big increase in total cross-sectional area. I'll bring up a graph in a moment, we'll talk about that. So the pressure is highest when the blood leaves the heart. There's a rhythmic rise and fall because of ventricular contraction. The further it goes away from the heart, the more friction there is, which leads to a decrease in the pressure. Arterioles have got a very large total surface area and a narrow lumen, therefore a significant drop in pressure. Capillaries have got a large total cross-sectional area, but they're incredibly thin. So again, we've got even more resistance to blood flow. There's a further drop due to the fluid being pushed out from the capillary into the tissues. Okay, this sort of graph comes up now and again. So this is the total area of each part. So this would be arteries, aorta, arteries, then arterioles, then capillaries, then venules, then veins, and then back into the vena cava. So you have velocity here, and then the cross-sectional area here. Now, a bit that gets lost on students, fairly sensibly, you can see that as the cross-sectional area increases, then the pressure falls, because the total cross-sectional area is increasing, but the cross-sectional area of each individual vessel is getting smaller and smaller and smaller as you go from aorta to artery to arteriole to capillaries. Now, then, when you leave the capillaries and go venules and then veins, the velocity of the blood increases even though it's at a very low pressure. And the reason for that is that you've got really big tubes in the veins. So you've got a big lumen, so you've got relatively less friction with the sides of the vein, so that you can have a big fat tube and therefore have low pressure but reasonably fast velocity. So the blood is being returned back to the right atrium and then can be pumped around the lungs again. Okay, that's it. I hope some of that made sense. If anybody has any questions, please feel free to ask them and I'll answer them for the next 10 minutes or so. There's a final Socrative quiz, which I'll just load up now. I hope you can all take part in that. So the next one. I'll just get the last Socrative quiz up. So the last Socrative quiz is running now. So if you haven't done the quizzes before, then please do go and take part. It's at socrative.com. You put in A-level biology tutor and please take part. There's 8 questions there. Next week it's respiration, aerobic and anaerobic respiration. Week after that, I think I'm going to go off on half term and go for a nice holiday in Spain. And the week after that, I'll start again. All of the details are on my website, which is alevelbiologytutor.com. If you've found this interesting, then please subscribe on YouTube. There are about 50 or 60 videos that I've made there. The ones going through the exam questions, exam papers appear to be particularly popular. Also, I'm on Twitter with Understand Biology. So if you want to follow me on that, you're very welcome to. Anybody got any questions at all, please ask them now. Or any points you wish me to talk about again for a couple of minutes before I go and have my tea. Can these videos be rewatched? Chloe, that's a very good question. The answer is yes, because I also stream them live on YouTube. So yes, they should be there, and hopefully I will be able to edit them to take out a few of the disasters. Thanks, that was really useful. That's very nice of you to say that. Thank you very much, Simran. Very helpful. Very nice of you to say that. What are the roles of the sphincter muscles? The sphincter muscles are a circle of smooth muscle, and they're around the arterioles. And when they contract, it reduces the diameter of the lumen, so it increases the pressure, so it reduces the flow of, so it increases the resistance, so it reduces the flow through that part of the arteriole. So you're going to reduce the flow of blood to the bit of the body which is downstream of that sphincter muscle. So the role of the sphincter muscles is to change in diameter and therefore change the amount of blood that can flow through. I hope that answers your question. Any more questions, people?